So I'd like to thank the organizers for allowing me to present my work to you today, in which I'm showing you how we are identified a pathway that regulates proportional growth of regenerating tissue. And this is not only important for those people who want to regenerate correct proportions, but also for people who are talking about tissue engineering. What is it? It's in the host animal or body that tells stem cells and progenitor cells stop growing and not to form tumors or become cancerous. So I use a zebrafish because it has such an extraordinary ability to regenerate compound structures, unlike us. So for instance, here is the jaw. They can lose completely part of the jaw and it will regenerate over time, including the bone, vasculature, and the innervation. Same thing for the heart. We could remove part of the apex of the myocardium. You'll get the blood clot, fibrosis, and then that fibrotic tissue will be replaced by myocardium. And same thing with the fins, the fish appendages, that they will, after loss, regenerate completely so that you can't see the difference between what was regenerated and what it originally had. And I'm using the zebrafish fin to try to understand how tissue regeneration occurs. So even though it has a different architecture than our limbs, it has pretty much the same tissues that we do, only just in a different pattern. And when it loses this tissue, it also goes through the same sort of events, clotting, immune response, reepithelialization, ECM remodeling, that we do. Only the difference is it doesn't induce scarring, but activates developmental tissue paradigms to restore the lost tissue. And this is what it looks like. So if we took at a regenerating fin ray, so one of the bone rays, and we took a cross-section, what we would see is this. So the bone rays are actually two juxtaposed hemirays, concave hemirays that you see I have depicted here. There's innervation, vasculature over skin, mesenchyme, connective tissue, and even joints. Another one gone. Right? And so the difference is that when it loses its appendage, it heals it rather quickly, and then this epidermis specializes and sends signals into the cells of the residual stump. These stump cells get recruited. See, these are adult cells, differentiated adult cells, to become progenitor cells. They proliferate and form what we call a blastema. And the blastema, in essence, is this proliferative cell mass that one could analogize as like a tumor. It's massively growing progenitor cells. Only the difference is the animal controls these cells so that not only does it grow the right architecture and size, put the tissues in the right place, but it grows to the appropriate size. And this gives us the ability to understand what is involved in an endogenous system, how you actually use these adult differentiated stem cells and go through some sort of reprogramming event to make them progenitor cells, and equally important, how do you control their growth so that the structure gets the right pattern and the right size. And so we came upon the third question, is how do you get the correct proportional size, by serendipitously when we were looking at immune response. So we looked at various immunosuppressants, and what we found is that those immunosuppressants that inhibit calcineurin resulted in the loss of this proportional growth control. So this proportional growth control can be done either by the fact that the animal just regenerates faster in the same given period of time, or it just loses the sort of boundary saying stop growing and it just continues to grow. And so we looked at this, and normally what happens in all tissues that involved in regeneration is you have a proportional growth. So your arms, limbs, liver, whatnot grow to the same proportion of the body. And here we go representing it as fin to body ratio, and this is always constant. So as the animal grows, its fin will grow, and it will grow in appropriate proportion. This is what we call isometric growth. However, when they lose tissue and they start to regenerate, they regrow tissue very quickly that's disproportional from the body, which we call allometric. So it grows back very fast, but somehow it knows at the right size and the right proportions to switch back to this isometric growth, which is proportional with the body. So then the question is how does calcineurin inhibition reflect upon this growth control curve? And what we see is that when we use different inhibitors to calcineurin, we basically lose this proportional constraint so that the animals continue to regenerate their fins well beyond the normal dimensions, right? Indicating that calcineurin is required for this proportional constraint that says this is when the right size has been reached, right? And we have no change in the overall body growth, so just with the regenerating fin. So then, as I mentioned, we were looking at immunosuppressants and how this might be influencing growth. And what we find is that if we use other immunosuppressants that either use the same mechanism, but due to the interaction have different targets. So this would target mTOR, where this would target calcineurin, or we use other immunosuppressants that target other immune response genes. We see that only when we inhibit calcineurin do we get this loss of growth suppression. So rapamycin, which affects mTOR, which is protein synthesis, has no effect on the proportional growth, or neither does cholesterol, which influences the NF-kappa B pathway, indicating that it's specific to calcineurin. So then the question is, well, how is it regulating this growth? So when we do cross-sections through the fin, so these are the regenerating fins, we see the overlying wound epidermis, the underlying growing blastema, and when we take measurements at the various distances of the thicknesses of these tissues, we see that there's no real difference in the size of the tissues or the blastema, indicating the growth is not due to an enlargement of a particular tissue type. When we look at the cell size, we don't see a hypertrophy, indicating that we're not influencing cell size to influence growth, but rather we're maintaining this proliferative growth at the tip of the fin. So we're maintaining cell proliferation. So then the question becomes well, if we're regulating the switch from going this regenerative growth back into this proportional growth, can we override an endogenous system of normally isometric growth control and switch it into a regenerative-like state? And the answer is yes. So what we find is that when we inhibit calcineurin, we induce allometric growth, so this regenerative-like growth, compared to the normal isometric controls. And this includes pretty much the paradigms or the genes that are involved in growth. And the nice thing about this is now we can actually look at the gene program that's involved in regulating proportional growth, because we can do it without having to have the injury response. We can induce regeneration growth without dealing with injury. So if inhibiting calcineurin is regulating the switch between regenerative growth and isometric growth, then the question is what does the endogenous protein's activity look like during these different growth rates? So we took tissue from different time points during regeneration, during the high allometric growth and as it slows down to the isometric growth, and then we looked at calcineurin's activity, phosphatase activity in the cell lysates from these tissues. And what we see is that when we have this high rate of growth, calcineurin's activity is low, and as we go back to isometric growth, its activity increases, indicating that calcineurin's endogenous activity is regulated as we go from a proximal to distal regeneration state. Right? So the two take-home messages here are that calcineurin seems to be acting as a switch to go from regenerative allometric growth into isometric growth, and that calcineurin's phosphatase activity is regulated during regeneration. So then the question is what is regulating it? So in the end, this gets us to one of the major mechanisms which involves body proportions altogether, right? So through all our developmental or those organs that do regenerate states, we have specific proportions with the body. And we know that from various animal models that there are local controls within the tissues, such as, for instance, oncogenes, tumor suppressors, etc. And we know that there are systemic factors within the animal that control also this proportional growth, so within the body itself. The question is that we don't have the full complement of what these things are, and we don't know how they're integrated so that you get a multi-tissued structure that grows proportionally. Okay, so then one of the things that we found out with fish is that position influences growth rate. So a group, Ken Poss's group at Duke Medical School, shows that if you cut proximally or cut distally, you have different growth rates. So a proximal cut will have a higher growth rate, and a more distal cut will have a lower growth rate. And these growth rates are coordinated so that irrespective of where you cut, either more distally or more proximally, they will be coordinated so that the fin will regenerate and reach the same proportional size at the same time. So then the question becomes, if we inhibit calcineurin, how do we influence this coordinated growth? And what we find is we basically lose it, so that irrespective of where we cut, whether distally or proximally, they grow at the same rate, so that we override or we lose this positional control of growth rate, so that one lobe cannot catch up with the other, as we have here. So then the question is, what molecular mechanism might be involved? Now, it was shown in the 70s and early 80s that retinoic acid is a major molecular mechanism which is involved in regulating positional information. So that, for instance, if we take these salamander limbs, which they also regenerate their limbs, that if you amputate at the wrist, you normally will get a regeneration of the complete hand from the wrist. However, if you increase retinoic acid signaling by increasing retinoic acid concentration, you'll start to proximalize the tissue so that from this wrist cut, you'll start to form, right, upper arm, lower arm bone, even more upper arm bone, and if you go higher concentrations, even a shoulder blade, upper arm, lower arm, and hand, showing you how important retinoic acid is involved in regulating this positional information. So then we looked at retinoic acid signaling in the regenerating fins after calcineurin inhibition, and we looked at the various factors that are involved in synthesizing retinoic acid from retinol, interacting with retinoic acid either to deliver it to the retinoic acid receptors, or it can be catabolized by retinoic acid CYPs, which basically turn it into hydroxyretinoic acid where it's no longer functioning. And what we find is those enzymes which are involved in promoting retinoic acid signaling goes up, and those that are involved in catabolizing retinoic acid and reducing its signaling going down. So we're basically regulating the whole retinoic acid, endogenous retinoic acid signaling system through calcineurin. These are two different inhibitors giving us the same information. So then the question is how does this fit? Okay, so we know that calcineurin is a phosphatase and that it is regulating retinoic acid, and the question is how? Well, we know as a phosphatases dephosphorylate proteins to either activate them or inactivate them, and therefore with the fish we have the advantages we have a collection of mutants which also have fin growth control problems, and then we can look at these and see whether or not they might be targets of calcineurin. So this is another long fin, which you can see has extremely growing long fins disproportionate with the body. And when we look at another long fin, we see that it has been found out that it's a potassium channel that regulates this disproportional growth. And what one finds is that mutations in this potassium channel increase intercellular calcium conductance so that you get this increased growth rate. So then we looked at this potassium channel to see whether or not it was a target. And we see that from the protein sequence, there's a putative docking site for calcineurin, and that it's surrounded by phosphorylation sites, which could in essence be regulated by calcineurin. So then we want to know, well, does calcineurin interact with this protein? And by doing co-IP experiments using the zebrafish protein in HEK cells, we see that using calcineurin, MIC, we can pull down the potassium channel. Here is showing endogenous calcineurin, and this is the pull down of the tagged version, and where we can actually see the interaction with the potassium channel, indicating that they do interact. So then the question becomes, what I do is I messed this one up. Oh, sorry. I'm actually missing a slide. Okay. So what we found is that now we see the interaction we actually tested in HEK cells. If we transfect this potassium channel with calcineurin, or inhibit calcineurin, do we influence the potassium conductance as we see here? And the answer is yes. So normally what happens if we increase the expression of this protein in HEK cells, we see an increase in potassium conductance. And if we inhibit calcineurin, we significantly upshift that, just like what we see in the mutant, indicating that the interaction between calcineurin and the potassium channel is functional. So then the question next becomes, well, if calcineurin is regulating its potassium channel, how is it regulated in order to regulate this? And we know that calcineurin can be regulated in different fashions. One is through calcium, when interacting with calmodulin to bind to calcineurin and activate calcineurin. And another one is there are endogenous proteins within cells that can inhibit calcineurin's activity. And so, I don't have this one either, do I? Oh, here we go. So what we see is when we do ratiometric analysis using dyes that bind to calcium, so that either when they're bound to calcium, they fluoresce when excited at 340, or they fluoresce only at 380 when they're not bound to calcium. And then we can take the ratio of this and see whether there's more bound or unbound calcium within the cells, in the regenerating fins. And what we see is that in the blastemas, where you have this high rate of growth, calcium levels are down, which would indicate an inactivation of calcineurin, so that fits the model. Whereas in the interray space, where regeneration is not taking place, so in differentiated tissue, it's up. So this indicates that calcium levels are changed during regeneration in a way that would regulate calcineurin, so that when you have this high rate of growth, calcineurin's activity would be down. Conversely, when we look at the Arcan, we see that at early stages of regeneration, it's highly expressed, and that its expression is downregulated as regeneration slows down, just very much fitting to the curve that we see low levels of calcineurin activity when we have high expression of this inhibitor, and increases in calcineurin's activity when we have low levels, indicating that not only is there ion control of this, but also transcriptional regulation in some way. So what I've told you today is that we're trying to identify, using calcineurin, how it is regulating positional information, or involved in the regulation of positional information and growth control. And I've shown you that it regulates a potassium channel, which indicates that bioelectrical signaling might be involved in regulating proportional growth and progenitor cell biology, and that calcium and that transcriptional regulation of an endogenous inhibitor is regulating calcineurin in a way that fits with what we observe in the endogenous system. And so we can use this now to look at what are the systemic controls or local controls that were regulating these factors in order to regulate positional information. So with that, I'd like to thank the people that were involved. So Satu, who is the postdoc that was the driving force of this, with the various master's students that contribute to the program, as well as people we collaborated with, Ying and Zhang at B-Cube, who was helping us with the calcineurin assays in the endogenous tissue, the genomics group, in which we're doing now, of course, deep sequencing to find out what is the transcriptional differences when we're influencing calcineurin's activity, and the fish facility, without whom we wouldn't get very far. So with that, I'd like to thank you, and I'll take questions.
Very nice work and so interesting. I'm just wondering whether you can educate us a little bit on the composition of the thing.
Oh, so, I mean, in essence, as I said before, it pretty much has many of the tissues that we have. So it has vasculature, peripheral nervous system, mesenchyme connective tissue. It has joints. It has bone, although the bone is ectodermal, so it's like our skull, not like the endochondrial bone that we have in our limbs. The skin has melanocytes that are the same cells that we have in our skin. It just has other pigment cells as well. So it pretty much, the only tissue that's missing is muscle, and that's at the very base of the fin. So.
Thank you, Christopher.
Oh, please. Maybe a stupid question, but I'm just curious. The fins of these fish, do they grow on forever, or do they sometimes stop?
So that's not actually a stupid question. So one of the things that actually people think that those animals that regenerate do maintain a continual growth. So if you keep feeding the zebrafish, it will continue to grow. It won't, I mean, of course, it has a fast growth rate during juvenile stage, and then it slows down, but it continues to grow, right? And even those, as I said, those other animals that regenerate, they seem to still maintain certain developmental paradigms in an on state, and they continue to grow. So one of the questions that people have is that those animals that do regenerate, is that because they are sort of already primed for it, because they have certain developmental programs on and that they're continually growing, or is it due to something else? And it's most likely a combination of both.
I think it's a related question. We know that some of animals or fish especially get bigger if they have more place to grow.
Mm-hmm.
So
Zebrafish are like that too.
And do you think this can be controlled also with calcineurin? Or is this controlled by this? What is the environment?
Well, no, I think so. What we see, for instance, we're still not certain how calcineurin might be influencing the rest of the body, right? So I don't necessarily know how calcineurin is. I can't say that if we inhibit calcineurin, for instance, that we are going to, stop the animal from completely growing. There's also that calcineurin has many other functions, right? So we will suppress the immune system, and that will kill them with time, right? And it also has other effects on other parts of the body, like the nervous system and whatnot. So it's a difficult experiment to test. But we know that there are, of course, hormonal influences on them, that the amount of food that you get, generally if they're in the tank by themselves, that means they get more food. It's not necessarily a space constraint, because they will beat each other up, right? And there will be a dominant one who will be big, and other ones which will be small in the same space. It's just because one will be able to get more food, just like one sees with rodents or whatnot. So it's not so much the space constraint, I would say, it's more the access to food that would control that sort of growth that you're talking about.
There's the other idea about the icebergs get much bigger because of the cold, and there's a relationship between surface and
Well, temperature, it would be more metabolic, right? I don't necessarily think that it would be more a general thing. These are isometric plachiotherms, and so they don't regulate their temperature. The surroundings regulate it for them. And in terms of, you're talking about surface in terms of tension within the tissues, or you're talking about?
It must be something, the relationship between, as you told, of growth, and in this case of body and outside.
Yeah, the environment. But I think that environmental thing is just simply access to food. So if they can get more food, because we feed them the same amount per tank generally is what happens. It makes it much easier for when you have thousands of tanks. And so a tank that will have 5 fish, the animals by default will get more food than a tank that has 30 fish. And so this is what I'm talking about, is it's more like the access to food.